Hello, everybody. This is Miss Peachy from your WCA biology class, and I'm going to be doing an overview of Unit 8, Lesson 2. The title here is Genetic Variation from Meiosis. So we are looking at the process of meiosis now, and meiosis is another type of cell division. Specifically, meiosis actually is cell division in only gametic cells or gametes. Now, what the heck is a gamete? Well, as you can see from our keyword here, a gamete is a sex cell. This is your egg or sperm cells. So these cells are unique because they are the only cells in your body that have half the number of chromosomes than any other cell. Because when an egg cell and a sperm cell, when they meet each other and they join, each cell brings half the number of chromosomes to create an embryo or to create a zygote in that case after fertilization that has all of the chromosomes necessary to make an entire individual, right? And so this takes place in the animal kingdom, it takes place in the plant kingdom, so it's not unique to just humans. It's all kind of multicellular living things, excuse me, or most multicellular living things do meiosis as well. So that's a type of cell division. It sounds a bunch like mitosis. I know the word is confusing, but it is different. And again, only ever in the creation of eggs and sperm. Okay? And it's important because reproduction is life. Life is biology. So this is all very relevant and vital and necessary to understand. So some of our vocab words here: crossing over, gametes, which we already defined, haploid, heredity, homologous, independent assortment, inheritable genetic variations, karyotype, and meiosis, which we also talked about as well. I will also talk about haploid right now. So half the number of chromosomes means you are haploid. So eggs are haploid and sperm are haploid because they have half the number of chromosomes that are found in a regular body cell. Okay? So in a regular body cell, if I plucked a big chunk of skin off me, that would kind of hurt. But if it did that and I looked at one of my skin cells under the microscope and had a very powerful microscope that enabled me to see the number of chromosomes inside of my nuclei of my cells, what I would find is I have 23 pairs of chromosomes. Now I inherit pairs because I inherit 23 from mom and 23 from dad, and they're pairs. So chromosome number 1, I have 2 copies of it. One copy comes from mom, one copy comes from dad, and they have the same genes on each of the chromosomes. So that's true for every single chromosome that you have, except for chromosome number 23 if you are a male. If you are a male, chromosome 23, you would have inherited an X chromosome from your mother and a Y chromosome from your father, so they would not be identical. They're never really identical, but they don't have the same stuff on them. Chromosome Y doesn't carry as many genes as chromosome X, but chromosomes 1 through 22 all carry the same genes in the same order, but there's different variations, there's different alleles. So perhaps you could inherit the allele for brown eyes from your mom, but you inherited the allele for blue eyes from your dad, and so you are heterozygous for eye color, big B, little b. So we'll kind of get into that a little bit more here as well. Let's look at the overview of meiosis, shall we? So the thing that has to happen before meiosis can begin is the cell has to first double the amount of chromosomes that it has. So in a human, we have 46 chromosomes, 23 of each kind, right? So 23 pairs. So I'm going to start off with, ooh, not a yellow pen. Good. 46 here, representing the total number of chromosomes we have. This is before meiosis. So before, then before meiosis even begins, we have replication that happens. So that when we begin meiosis, we have a cell that has 92 chromosomes. Okay, that's important. So meiosis is oftentimes called double cell division. Double division of cells here. So the first division is known as meiosis 1. Meiosis 1 is where we take that 92 and we divide it back down into 46, but we have two cells now. Right? Then we have a second division, and that second division is creatively known as meiosis 2. I'm going to write this just a little smaller because it's interfering with my, so I'm just going to, there we go. I just got to write small. It's very hard with this big pen. And that divides this into 23. This is again for the human, because not every organism has 46 chromosomes, right? So for the human, we end up with, from our original 46, 4 cells, each with 23 chromosomes. For the sake of being able to draw a picture with a little squiggly tail, I am going to say these are all sperm. Because remember, the whole purpose of meiosis is to make egg cells or make sperm cells. Now, I have egg cells personally, but they would just be a circle. And I just didn't want to just draw a circle. I want to draw a circle with a little squiggly tail. So we're going to call these sperm cells just so that you guys get a visual that these are gametes here, right? That this is the process that's making gametes. So we made some gametes. Yay. So my meiosis has 2 divisions, double division, first replication, then double division, that results in 4 cells where once you had 1. These cells down here are all haploid cells. All the sperm are haploid cells, all the eggs are haploid cells because they have half the number that we started with. That's why. So let's say we follow a couple of genes. So for eye color, let's say I had, or not me, but whoever this is, has the eye color big B, little b. They inherited the big B allele from mom. They inherited the little b allele from dad. When they replicate, they now have 2 copies of this, right? These big B, little b are known as sister chromatids because they are connected to each other. They're a part of the same chromosome. These are all the same gene. So altogether, these are known as homologous chromosomes. So, this set here is homologous to this set here. So we have homologous chromosomes here. During meiosis 1, the sets of homologous chromosomes, the sister chromatids stay together, but the homologous chromosomes go their own way. So one of my big B's ends up over here, and one of them ends up over here, right? During meiosis 2, the sister chromatids will separate. So each sperm ends up with only one B. If this sperm goes on to make a baby, that baby is going to inherit the allele for brown eyes. If this sperm goes on to make a baby, that baby will inherit the allele for blue eyes. So there's some inheritable genetic variation depending on which allele is inherited here. The other thing to note here is that we have way more than one gene, right? So if we have another gene in here, just throw up another one just so we can kind of follow it. So also have big A, little a. Again, during replication, we make 2 copies of that. During meiosis 1, sister chromatids stay together, but homologous chromosomes separate. But during meiosis 2, those sister chromatids separate, and they don't have to follow the same path as the B's did. So big B, big B went here. I have no pointer here. This is driving me nuts. Big B went here, right? Big A doesn't have to go there. Little a could go there, and big A could go here. They don't have to follow the same path as other genes did. They still have to sort, so you're only going to split them up. One goes to each one, but where they go is independent of any other allele, any other gene. So this is called independent assortment. This is one of Mendel's basic principles. And that just means that every gene you have, when it goes through meiosis, it just randomly gets sorted into an egg or sperm. You make 4 eggs or 4 sperm from every cell, and which of your sister chromatids ends up in them is kind of random. And that's what independent assortment is. So that creates some variation too, right? So now you've got 4 different things here. You've got one that has big A, one that has big B little a, one that has little b big A, one that has big B big A, and one that has little a little b. So there's 4 potential possibilities as far as what traits can be inherited by the offspring. So that's kind of cool too. So the other way in which inheritable genetic variations is introduced is through something called crossing over. And this happens within the stages of meiosis. So meiosis 1 and meiosis 2 also are divided into 4 stages: prophase, metaphase, anaphase, and telophase, just like you learned in mitosis, right? And that basically just shows us like what's happening inside the cell, that the nuclear membrane is dissolving, that the chromosomes are shortening and thickening, that they're moving around and they're being divided onto different sides of the cell, and the cell is pinching off in the middle, and you have cytokinesis, and all of those stages happen in mitosis too. Two meaning TOO, meaning also, in both stages of mitosis. However, in meiosis. In meiosis 1, it's super crowded. So the first thing before meiosis 1 could happen is that the cell had to replicate all of its chromosomes. So where once there was 46, we now have 92. It's like ridiculously crowded. When it gets that crowded, the chromosomes can overlap. So homologous chromosomes can overlap right here. So think of this as your big B, little b, and this is your other big B, little b, right? We saw earlier. And they're the same genes. So it's not like you're overlapping the gene for eye color with the gene for hair color and you end up growing hair out of your eyes. That doesn't happen. What's happening here is the genes that represent eye color, you've got 2 sets now of 2. So you've got 4 alleles, and they get kind of tangled together. And so then you end up with like a hybrid here at the end where little bits of each kind of get attached to the other ones. And this process is called crossing over. And the process of crossing over only takes place in prophase 1 of meiosis 1 because it's super duper crowded. And so that's when it happens. This whole structure here is known as a tetrad. And it's just because there's 4 of them. And so this is another way in which I'm trying to be dramatic, but it's not working, in which inheritable genetic variation gets thrown in there. So you can have variation just because of independent assortment, and you have variation because of crossing over. And so what that means is that the end result, those 4 egg or 4 sperm that form, are different from the one that started. Genetically, these are different than the original. Meiosis leads to differences. Mitosis leads to sames. So that is this lesson in a nutshell. Hopefully it helped you out a little bit, made some sense to you, and you understand meiosis a little bit better. If you have questions, let me know. Otherwise, I will see you in the next one.